Okay, so thanks everyone. Today we're going to hear from our PhD student, Rebecca Degnan, and she's going to be talking about some of her PhD work on harnessing double-stranded RNA for myrtle rust control and biological insight. So if you just want to go to the next slide. So I'm Dr. Anne Sawyer. I'm a research fellow here at QAFI at the Centre for Horticultural Science. And I'm also jointly appointed through the School of Chemistry and Molecular Biology. And so Beck is actually one of my PhD students, so I'm very happy to welcome her today. So if you want to go to the next slide, Beck. But first, I would just like to acknowledge the traditional owners and their custodianship of the lands on which we meet. And I pay my respects to their ancestors and descendants who continue cultural and spiritual connections to country. And I recognise their valuable contributions to Australian and global society. Next slide, please. All right, so this seminar is scheduled from 12 until 1. And at the conclusion of the seminar, we will hold a Q&A session. So if you have any questions, please just put them into the Q&A button at the bottom of the screen, not the chat button. And then we'll go through the questions at the end of the seminar. Okay, next slide. Okay, so as I mentioned, today we're going to be hearing from Rebecca Degnan, who's a PhD candidate. She works across QAFI, SCMB, and the Department of Agriculture and Fisheries. And so Beck is actually highly awarded. She's a Reitman Scholar with PBRI. She's the co-founder of the Plant Health Student Network, and she was recently recognized as UFresco Scientist of the Month. And as I mentioned, her work is on myrtle rust, so developing double-stranded RNA sprays to control and also understand myrtle rust. And she's recently published this work in molecular plant pathology and also in communications biology. So take it away, Beck.
Thanks, Anne, for the introduction. And thanks to QAFI for having me and everyone for tuning in today and taking time out of your lunch break. So I'd just like to begin by acknowledging the Yagra and Turrbal people, who are the traditional owners of the land on which I live and work, and the land on which this research was conducted. And also, as a scientist, it's important that we acknowledge that Indigenous sciences have been preserving and protecting this land for over 60,000 years and also continue to do so today. So as Anne mentioned, today I'm going to be talking about using double-stranded RNA to control rust fungi and also to study rust fungi. And I'll talk a little bit more about what I mean by that later on in the presentation. So my kind of rust of interest and the main rust that I'm going to be talking about today is Austropuccinia psidii, and it's the causal agent of myrtle rust. So many of you may have heard of myrtle rust before. It infects plants of the family Myrtaceae, and we actually have around 2,000 species of Myrtaceae that are native to Australia. So there's a lot of kind of scope for damage there. This family includes a lot of those really iconic Australian genera like eucalyptus, melaleuca, which is paperbarks, and psidigiums, which is our garden lily pillies. And the symptoms have a real range in severity. So you can see kind of anything from relatively minor leaf spot and blight and pustule formation to this kind of whole tree malformation and mortality, like you see in this image on the bottom right here. Many of these hosts that I've mentioned, they have high cultural, environmental, and economic significance. So in terms of cultural significance, we can be talking about things like seasonal indicators, building materials, or even food. Conservation or environmental significance. A good example is the broadleaf paperbark, which is a keystone species in kind of coastal wetland type environments. And yeah, in some parts of Queensland, it's getting pretty smashed by myrtle rust. And then in terms of economic importance, an example that we see again here in Queensland is the lemon myrtle industry. So at the moment, we're seeing in untreated lemon myrtle plantations yield losses of up to 70%, which is pretty huge. And in some cases, growers actually have had to move their entire plantations to kind of remedy this. And yeah, as a result of kind of the population decline and localized extinctions that we see as a result of myrtle rust, we now have over 50 species listed as conservation priorities. And further, a study by some researchers at the School of the Environment at UQ a couple of years ago, which actually surveyed over 1,000 Myrtaceae populations in Queensland and New South Wales, they found that 16 species of Myrtaceae of endemic rainforest trees will be extinct within a single generation, and a further 20 will be at risk, all because of myrtle rust. So I hope that kind of gives a bit of overview of how damaging myrtle rust can be and why we need to do something about it. So Austropuccinia is an interesting fungal pathogen and an interesting rust. It has 3 main spore stages: the Uridinea spores, Telia spores, and Basidia spores. And it can reproduce both sexually and asexually, although the kind of second half of this life cycle is still being untangled. And it's really important to note here that this is not the same biology as the model rust, Puccinia graminis, which has an alternate host. So myrtle rust is not a model system. It's a tropical rust with these long-lived perennial hosts, and it can complete its life cycle on a single host. So all of these things have really important implications for the management of the pathogen. And existing controls of rust, including agrochemicals such as fungicides and breeding for resistance, and then deploying these resistant genotypes, are not alone sufficient for myrtle rust management. They tend to be either short-term solutions, or they're limited by evolving pathogens or financial and environmental costs. And it's not to say that these controls are not effective at all, but they're just not alone effective. So we have some other really exciting developments happening in the myrtle rust management space. So there's a lot of promise in seed banking and cryopreservation and tissue culture, which you may have heard about previously from Alice Hayward and her group. But yeah, it's kind of this really complex and pretty destructive pathogens. So these complex destructive pathogens really require these multi-pronged approaches, and that's what we're hoping we can contribute to here. So before I kind of dive in, I just want to take a step back out and look at rust fungi as a group. So rust fungi are a huge group of plant pathogens. I think there's nearing 10,000 described species. So they're like, it's a massive group. And there's basically a rust for like any kind of plant that you can think of. So there's agricultural rust, like soybean rust, wheat rust, and coffee rust. You have your environmentally significant rust, like myrtle rust, and there's also white pine blister rust in North America. And then you have your rust of ornamentals. So that can be things like frangipani rust or monstera rust. And you may have seen frangipani rust around you recently. It's everywhere at the moment. But yeah, the frangipanis get totally smashed by frangipani rust. So I think the kind of size of this group and also the scope of what rusts can impact really highlights the need for effective management, and it also highlights the possibilities for effective management. So this is where we think double-stranded RNA-mediated control can come in and contribute to this arsenal of management tools. And when we talk about double-stranded RNA-mediated control, the hypothesis for the mode of action is RNA interference, or RNAi. So RNAi is a form of post-transcriptional gene silencing. It's largely conserved throughout eukaryotes, but like with everything in biology, there are a few exceptions to this. So in this mechanism, endonuclease known as DICER recognizes long double-stranded RNAs, or hairpin RNAs, and it cleaves these dsRNAs into small interfering RNAs, a much shorter fragment. These siRNAs then complex with an Argonaute protein to form the RNA-induced silencing complex, or the RISC, and then RISC guides sequence-specific degradation of homologous mRNA. So what that means is that this RNA-induced silencing complex is basically looking for mRNA that has a sequence that matches this original piece of long double-stranded RNA or hairpin RNA. And once it finds that bit of mRNA that matches, it's going to degrade it, and that leads to silencing of target genes. And in some eukaryotic organisms, you also get this amplification by RNA-dependent RNA polymerases, and that can lead to this kind of continuous looped silencing of the target genes as well. So it's pretty cool because we can actually co-opt this mechanism to defend plants against a variety of plant pathogens, including fungi. And this can be done through a non-GM approach referred to as exogenous double-stranded RNA. There are a few different names for it, but that's how I'll refer to it. So in this approach, pathogen-specific double-stranded RNA is applied directly to the plant, usually via spraying. And then the hypothesis is that double-stranded RNA is then taken up into the plant or fungal cell, and then this process is triggered. So double-stranded RNA can also be complexed with a variety of carriers for nursery, ex situ conservation, or field applications in pathogen management. And the example of this that many of you might have heard of already is the complexing of double-stranded RNA with the layered double hydroxide sheets to form the bioclay complex, which is the work of Nina Mitter and her collaborators. So basically, before we just kind of sped straight into field trials or working out if we can use double-stranded RNA in the field or something, we needed to firstly know if Austropuccinia was actually going to be amenable to double-stranded RNA. Like with any control measures, some work better in some pathosystems and some work better in others. So there's no guarantee that it's going to work, and you need to actually do the kind of more basic stuff first to actually figure out if this system was going to be amenable to double-stranded RNA. And luckily, a couple of my supervisors, Alistair McTaggart and Louise Shuey in particular, have worked on Myrtle rust for a really long time now. So we had this kind of really solid understanding of how this system worked, and that was a really good starting point. So I'm going to firstly go through the results from this study and tell you about what we found. So the first thing to do was to look at the RNAi machinery. Basically, we need to know if the genes are actually present, because like I said before, this pathway is largely conserved, but it's actually not always conserved. So the sister group to the rust fungi, which are the smut fungi, the Eustelago-mycotina, hopefully I said that right, their entire subphylum actually doesn't have any of these genes. And they're pretty closely related to the rust. So we needed to actually check in the rust fungi and make sure that these genes were present. So we found that the main RNAi pathway genes, so the RNA-dependent RNA polymerases, the diaceres, and the argonautes, they were all conserved across 8 families of rust, including the family that contains myrtle rust. And exogenous RNAi has also already been shown experimentally in a 9th family, in the Phacospiraceae, which is the family that contains soybean rust. So we knew that the machinery was present, and then we needed to kind of work out if it was actually going to be functional. So before kind of going straight into just spraying double-stranded RNA on the plants, we wanted to make sure that the spores were actually going to be able to take up the double-stranded RNA. So here we use Cy3 labeled double-stranded RNA and showed that the Austropuccinia uridinea spores take up double-stranded RNA really early in germination. So these 2 panels here, A and B at the top, those are Austropuccinia uridinea spores, the main infecting spore stage. And the glow that you're seeing is the double-stranded RNA being taken up into the germ tubes. And we did use a nuclease to ensure that any of that glow we're seeing is actually on the inside of the spore and not just sticking to the outside or something. And then what you're seeing in panel C here is actually the teliospores of Austropuccinia. So interestingly, they don't take up the double-stranded RNA. We're not exactly sure why. It might be something to do with their spore morphology, like a thicker wall or something like that. But yeah, they don't take up the double-stranded RNA. And we also had a look at uptake in a different rust species, in Coliosporium plumeriae, which is the causal agent of frangipani rust. And you can see from this second set of images that the double-stranded RNA is taken up very early on, again, looking at panel A over here down the bottom. And you can see that it's already glowing, even though all we've got is this little germ pore. So that is super early in germination, hasn't even developed that germ tube yet, and already it's taking up the double-stranded RNA really readily. So the next thing to do was to actually choose which genes we wanted to target and then design and synthesize our double-stranded RNA. So targets had a range of functions, from essential housekeeping genes to genes involved in haustoria development. The haustoria is this little structure down here that obligate biotrophic pathogens actually set up inside the plant to obtain nutrients. So we also targeted genes expressed in that. And then we used a variety of assays to basically test our hypotheses. And we use these detached leaf assays to do kind of initial screening of all of our targets. We screened 11 targets initially. Then we set up these in vitro assays on artificial leaves to visualize the impact of double-stranded RNA on rust infection physiology. And then we also developed an in-planta assay on roughly 1-year-old Syzygium jambos, which is rose apple, to kind of test our double-stranded RNA treatments in a more applied setting. So starting firstly with the results of the detached leaf assay, and what you're looking at here basically is each of these pairs is one control and one treatment. So the control being the negative double-stranded RNA and the positive being the plus double-stranded RNA, and they're all different targets. And the reason that we do these detached leaf assays in pairs is so we can take the two leaves, the two opposite leaves from the same node. So they're from the same node on the same plant, same age, growth stage, everything. It should minimize all other variables and allow us to have a really good comparison in a control versus treatment. And then all of the ones that are in green were a significant reduction between the control and treatment, and all the ones in this yellowy orangey color were not. And then GFP is included as a non-specific control. So that's basically just to ensure that double-stranded RNA doesn't have some kind of like inherent toxicity. And if we're targeting a gene that's actually not found in our pathogen, it's not going to have any effect. So we would expect to see no difference between the control and treatment in our non-specific control. So our detached leaf assays showed that application of 8 out of our 11 double-stranded RNA treatments resulted in a significant reduction in pustule development. The targets, when targeting the genes that were highly expressed in the haustoria, they were a bit less effective. I think a couple of them were these non-significant ones, and one of them was kind of just barely significant, but they were definitely a bit less effective than the really highly and constantly expressed barcoding gene targets. So we also made a couple of different constructs targeting the same gene but different lengths, just to kind of compare efficacy of different size double-stranded RNAs. And what we found supported what was already in the literature, which is that constructs in that shorter range of 200 to 400 base pairs were more effective than the longer ones. So continuing on to the in vitro assays now, we just picked the top 4 targets here to continue forward with. So in vitro, and this was on these artificial leaves that are, they were designed or innovated by our collaborators in New Zealand, a team of kind of biologists and chemists and engineers. They're PVP-coated PDMS discs. And the really cool thing about them is that if you germinate Austropuccinia spores on agar, you'll only see a germ tube. But if you do it on these artificial leaf discs that are meant to kind of mimic the tactile properties of a leaf, you actually can see much more of the infection physiology, as I'll show you more in the next slide. So we saw a significant reduction in germination rates in all 4 of our treatments in vitro. And I'll just show you what the infection structures look like before we compare them in the different groups. So this is kind of a germinating spore. This is the germ tube here. This is what we call cytoplasmic streaming, which is, I'm actually not sure if you can see my clicker. Well, in this picture on the right at the top here, this is cytoplasmic streaming. So this is the contents of the spore being pushed out through the germ tube. And then in this aprosoria formation image here, what we're seeing is that contents kind of aggregating down the end of the germ tube, and that's going to then swell up to form the aprosoria. And then the infection peg forms out of the aprosoria, and that's actually the piece of the fungi that is going to penetrate into the leaf and then set up all the feeding structures and everything. So when we look at compare the infection physiology in vitro, you can see in the negative control and the non-specific control, we're seeing these beautiful aprosoria and infection pegs forming. This was really cool for us to see. Like, even some of my supervisors who had been working on myrtle rust for a while, they hadn't ever really gotten to see it like this before, because you just can't really visualize it on agar in the same way. So very cool to have access to these artificial leaves. And then if we look at our treatments, either we were seeing just like very few germinating spores at all, or we were seeing these kind of short, stumpy germ tubes. In some cases, we were seeing these long, withered germ tubes. But yeah, we were never seeing anything that resembled an aprosoria or an infection peg in the same way that we were in the control. So the really cool thing here is that even when these spores were germinating, those germinating spores are never going to result in an infection, because they're just not producing the physiology that they need to actually infect a plant. So yeah, that kind of showed us also that the double-stranded RNA doesn't need pre-processing by the plant. Like, we'd already seen that it's taken up directly into the spores, and then it's clear that the spores can also actually process it and do something with it themselves without it needing to be processed by the plant. So we also repeated this assay in the frangipani rust, and we saw very comparable results. So we saw even less germination here. I've tried to capture in these images the spores that we did see germinating, so you can see what they looked like. But either we saw this kind of short, stumpy germ tube happening, or these really weird, knobbly ones, and just, yeah, very long and withered. But yeah, also important to mention that this, of course, involved designing new double-stranded RNA constructs, because the constructs are highly sequence specific. So you can't use the same ones between different pathogens, which is actually a big perk of the system, that it's so highly specific. So then to wrap that all up, we wanted to do a bit of an in-planta experiment on whole trees, just small trees, but this is on rose apple, as I said previously. So we just co-inoculated via spraying our Austropuccinia spore inoculum and the double-stranded RNA treatments or controls. And again, we saw a significant reduction in disease coverage on the leaves at 2 weeks post-infection. So in the case of one of our treatments, we actually didn't see any rust at all. And this was a pretty, infections at this time of year were pretty chill, like they weren't too extreme. You'll see in some later images, like at some times of year, you get much more extreme infections. Yeah, so this was a relatively chill infection, but we were seeing really good inhibition of disease in planta as well. So this kind of answered our question altogether. Is Austropuccinia amenable to double-stranded RNA-mediated control? That yes, it looks like it is. We also did some other work in this paper looking at kind of dose of double-stranded RNA and efficacy like related to dose. So if you're interested in that, feel free to go and check it out or get in touch. But yeah, we kind of could show after this that this was going to work well in this system and it was worth pursuing further. So the next logical step for us was to basically find out if double-stranded RNA could also be used preventatively and curatively against metal rust. Obviously, if you're spraying a treatment in like a field setting, like a plantation maybe, or in a nursery, you're not going to be co-inoculating your treatment with your fungal pathogen. Like it's always going to be before or after. And especially that curative aspect, that's super important. Like growers can see there's disease there and then go and do this treatment. So if it's going to be used in an applied way, it's really important to know if it can actually be used preventatively and curatively. So we started with a preventative experiment where we applied the double-stranded RNA at 48 hours pre-infection. And when plants were sprayed 2 days prior to inoculation, we saw this significant reduction in disease coverage at 2 weeks post-infection once symptoms had developed. And in addition, we also used some scanning electron microscopy in these images on the right here to sample treated and untreated leaves. And we did that at 2 days post-inoculation. So that was 4 days post dsRNA application by that point. And you can see in this panel on your right here that in the control, the spores germinated into these really long germ tubes and established these aprosoria, which you can actually see kind of plugging into the leaf here, which was super cool to see. And in the treatments, the spores were actually majority ungerminated, but when they did germinate, we saw these really kind of short, stumpy germ tubes that are just not really getting anywhere at all. And I've included this little diagram up on the top left here to just show kind of what the pathogen would be doing within the plant at that time point. And I've kind of carried these diagrams throughout the next several slides so you can see what the pathogen would be doing at the time of double-stranded RNA treatment. So for curative treatments, we firstly trialed a spray at 24 hours post-infection, and this image on the left here shows what the plant looks like at 24 hours. So at this stage, pustules have germinated and penetrated into the leaf. You might be seeing some haustoria being set up as well, but you're not seeing any visual symptoms on the leaf surface. It's all just happening internally at this point. And in terms of our disease assessment, at 2 weeks post-infection, we again saw a significant reduction in disease coverage on treated plants, as you can see here by the lack of pustules and also reflected down in the disease coverage graph as well. So we also wanted to know kind of how the plant was going to respond in a little bit more of a long-term setting. So it's important to know that the plant is actually going to bounce back and continue to do well, and not just do well while the double-stranded RNA is like sitting on the leaf. So we wanted to look at the kind of plant health at 6 weeks post-infection. And we chose that 6-week mark because that should be plenty of time for a plant to kind of recover and have lots of nice new growth and things like that. So we developed this qualitative plant health scale basically to assess if a plant's alive, has it grown, and how much has it grown. And then we just gave each plant a ranking from 0 to 5 based on how well it was doing. And again, we saw a significant improvement in plant health in our treated plants compared to our control plants at 6 weeks post-infection. I'll also mention we did do some kind of plant physiology work looking at the photosynthetic capacity as well, measuring stomatal conductance and things like that. And I haven't included that in this talk, but if you're interested, feel free to go and check it out in the paper. We then extended this curative window out to 6 days post-infection. So this is what the plants look like at 6 days. You're starting to see the first symptoms of metal rust on the plant. So this red spotting that you're seeing is actually the plant responding to the metal rust by producing anthocyanin. And yeah, that tends to be the first symptom that you actually see on the plant. So this is when they were receiving their spray, and at 2 weeks post-infection, again, we saw a significant reduction in disease coverage on treated plants, and it looks a little bit different to the 24-hour curative treatment at 2 weeks, because of course, by the time the plants are being sprayed, they already have this red pigmentation on the leaves. So that's not going to just disappear. But our treatment did seem to be kind of preventing the infection from progressing further, which is really cool. And then if we look at 6 weeks post-infection, again, we see this significant improvement in plant health. And this is where I kind of got pretty excited, because if you look at the plants, some of them have actually defoliated even in the treatments. So they might just not have, we might have not quite gotten it early enough for it to fully save the leaves that were already there and already infected. But what's really awesome is that they're bouncing back really well. So they have this beautiful new growth up here and down here. And they were doing much, much better than the controls, which either had no new growth at all after defoliation, or like this one on the bottom left here, it had a tiny bit of new growth and it was just immediately reinfected with myrtle rust. So we weren't seeing any reinfection in our treatments, only in the controls. And yeah, that was pretty special to see, I think, because it kind of lends itself more to like a true applied potential for the treatment. So we also did a little bit of microscopy at this 6-day curative time point. So we sampled for microscopy at 8 days post-infection, which was 2 days post dsRNA treatment, and that was just to give the double-stranded RNA time to do something. And we did some 2-photon confocal microscopy and also some scanning electron microscopy. So in the 2-photon microscopy, which kind of allows you to visualize quite deep into the leaf, you can kind of go through all the cell layers in the plant and kind of see what's going on. We were seeing a lot of these IH is intercellular hyphae, and then the H is haustoria. So we're seeing a lot of intercellular hyphae and haustoria populating the plant cells in the controls. And then this is a scanning electron microscopy image on the right here. And yeah, they're typically black and white, but I've just colored this one in for visual impact, I guess. But yeah, basically you can see how the fungus actually is like really just ruptured through the leaf cuticle. So this is how the infection process goes. Like the pathogen penetrates into the leaf, it sets up all its feeding structures and stuff. And when it's ready, it penetrates, like erupts back out through the leaf cuticle. And then there's thousands of new Uridinea spores, and then they go off and settle their own infection. So it's a pretty daunting image, I think. And then if we look at our treatments, we're still seeing the intercellular hyphae and haustoria inside the plant, which makes sense because those would have been set up days already before the double-stranded RNA treatment. And we had some beautiful images here of haustoria that were pretty exciting. And maybe it is a little bit less fungal material inside the plant than the controls, but this was difficult for us to quantify. But the cool thing is that if you look on the leaf surface, you could see all of this kind of evidence of where the fungus had started to erupt through the leaf cuticle, and then it's like it had just been stopped in its tracks. So like closed up, the spores had kind of stopped developing, or they were kind of all shut in there, like you can see in this image. And that was really further evidence by what we were seeing, you know, just with our eyes on the plants. Like it really looked like the disease had been stopped in its tracks. So then we wanted to extend the curative window out to 2 weeks post-infection, or 14 days. So this is what the plants look like at 2 weeks. This is when we would normally do our disease assessment. So it's a full-blown infection by this stage. Obviously, you can't do the 2-week post-infection disease assessment because they're already out 2 weeks and they're already covered in disease. So instead, we just looked at the qualitative plant health at 6 weeks post-infection. And again, we saw this significant improvement in plant health in our treated plants. And in this case, in the case of the 14-day curative treatment, most of our plants had experienced some degree of defoliation. You can see it in this beta tubulant image on the stem here, and in the EF1A, it's a little bit more hidden, but it's kind of underneath this leaf on the bottom branch. And what's really nice is that they were just recovering so well. So there's definitely a little bit of variation in how well the plants recover, but overall much better than what we're seeing in the controls. So just a little bit of a visual recap. This is all of our controls at 6 weeks post-infection that did not receive the double-stranded RNA treatment. And this is all the treatments that did receive the double-stranded RNA treatment. And yeah, they just look much happier. I don't think there's too much more I need to say for this set of images, but yeah, so basically we did this preventative treatment at 48 hours pre-infection, and we saw a significant reduction in disease coverage at 2 weeks post-infection. We also did curative treatments at 24 hours, 6 days, and 14 days post-infection, and we saw a significant reduction in disease coverage, and we also saw an improvement of plant health at 6 weeks post-infection. So this kind of answered our next question and showed that double-stranded RNA can be used to prevent and cure severe myrtle rust infections in mature plants. So for the last little part of this seminar, I'm going to kind of move away from using double-stranded RNA as a control for myrtle rust, and I'm also been really interested in kind of the fundamental biology of rust fungi, and we wanted to see if we could utilize double-stranded RNA kind of in the place of traditional gene editing techniques that are just not viable in this system. And yeah, see if we can answer some more questions about kind of the fundamental biology. So the element of this research that I'm going to share today is all about effectors in Austropuccinia. So of course, control measures like exogenous double-stranded RNA and really any control measure for a pathogen is going to be most effective in the long term when used in collaboration with other approaches, but also pathogen management is generally improved by developing our understanding of the pathogenicity strategies that pathogen uses. So talking about effectors, when a fungal pathogen infects a host, it sets up this hostorium inside of the plant cell, which I've talked about previously. And this functions both as a feeding site through which the fungus obtains nutrients, but it also acts as a site to secrete avirulence proteins known as effectors. And these effectors kind of facilitate infection by interfering with plant defenses, amongst other things. So gene for gene theory says that disease resistance is conferred through this kind of compatible interaction between an effector encoding avirulence gene in the pathogen and a corresponding resistance gene or R gene in the host. So this in turn triggers this kind of co-evolutionary arms race. And in this arms race, the pathogen recognizes the microbe-associated molecular patterns and it attempts to defend itself first through this pathogen-triggered immunity and then through effector-triggered immunity. And that's what you're seeing in this zigzag model here. So since Austropuccinia was only introduced about, that's 14 years ago now, Australian Myrtaceae haven't actually undergone co-evolution with Austropuccinia. And that's one explanation or possible explanation for the really wide host range. And excitingly, a collaboration with Grant Smith and Louise Shuey has actually identified some candidate effectors in Austropuccinia through a time course RNA-seq experiment. So since effectors are really, they're super fast evolving, they can be highly variable within the genome, they're often members of these large gene families. Yeah, all of these things can make their functional characterization really difficult, especially in non-model systems and especially in an obligate biotrop where you can't even stick it on a culture plate. And this is where double-stranded RNA can come in and hopefully help us to learn a little bit more about the fundamental biology of this pathogen. So, like I mentioned, our collaborators have identified two putative effectors that are expressed at a really early time point in infection. And basically, we wanted to know if these are true effectors and also what is the role that these putative effectors play in infection. I apologize in advance for saying putative effectors so many times. So we started by designing and synthesizing double-stranded RNA to target these two putative effectors and using double-stranded RNA-based assays to inhibit them both in vitro and in planta. We saw that putative effector 2 inhibited infection in vitro and in planta. However, putative effector 1 did not inhibit infection in vitro, but did in planta. And this one is really of interest because this is what we would anticipate to see for a true effector, seeing as effectors are typically expressed during this compatible interaction I talked about before with a host plant. And as a result, we would not expect the inhibition of an effector in vitro to have actually any impact on germination itself at all. So yeah, because this putative effector 1 was matching the kind of expected effector profile, we wanted to look into it a bit further. So we sequenced this putative effector and found that there were actually multiple variants present. So we cloned out this effector 1 so we could separate out the variants and ensure that we could actually synthesize double-stranded RNA for our assays that was targeting those specific variants and not a kind of mix of the multiple variants. So when targeting the RD19 variant, we saw inhibition of germination and infection both in vitro and in planta, suggesting that probably not the variant that we're after. But when we targeted RDO1, we saw that it did not actually inhibit the germination in vitro, but it did in planta. And that kind of puts it forward as a really promising candidate for a true effector. So as I said earlier, we also know from the time course RNA-seq experiment that these genes are expressed early in infection. And we know that expression drops dramatically between the 24 to 48 hour mark. So this, along with our previous double-stranded RNA-mediated inhibition assays that I've just showed, suggests that the putative effectors are essential for early stage infection, but potentially are not needed to sustain infection, or in other words, they're maybe not essential for that late stage infection. So we hypothesized that the curative application at 3 days post-infection of double-stranded RNA targeting the 2 variants of this putative effector 1 would have no significant impact on disease symptoms in planta. And we included beta-tubulin, which is one of the treatments that we used in the previous curative experiments. And we just included that as basically a positive control to ensure that the double-stranded RNA was acting, or was able to be acting curatively, as we had seen previously. So we saw that beta-tubulin double-stranded RNA did significantly inhibit disease coverage in planta. And we also found that dsRNA targeting these 2 putative effectors had no impact on disease coverage when applied at 3 DPI, at which time, 3 days post-infection, at which time these genes are not significantly expressed. So that kind of matched our hypothesis for this assay. And all of this is working towards supporting the hypothesis that putative effector 1 is a true effector in the Austropuccinia pathosystem and is crucial for early infection, but not for sustained infection. So to kind of summarize what we've done so far in this effector work, this is still in progress or in preparation, so it's not quite wrapped up yet. Yeah, we've examined 2 putative effector genes in a rust fungus, which are typically specialized pathogens, to determine kind of whether its effector landscape may actually be facilitating its behavior as a more generalist pathogen. And double-stranded RNA-based assays show that these putative effectors are crucial to cause but not maintain an infection, and inhibition of variants belonging to a putative effector with high intragenomic variability, so that's that putative effector 1, cause these differential expression of symptoms in vitro. And this might indicate that the expression profile of this gene family is actually under a strong diversifying selection, but that's kind of what we're looking into at the moment. And to learn more about the mode of evolution, or the type of evolution of these effectors and how this might influence pathogenicity and host range of Austropuccinia, we really want to look at the synonymous versus non-synonymous substitution rate of these effectors, as well as the kind of genetic landscape, meaning like what's around these effectors, what's like sitting on either side of them, and also timing of expression and things like that. So yeah, that's where we're at currently with the effector work, and we're also doing a few other things in the kind of fundamental biology space, looking at transposable elements as well, and hopefully I'll have more to share on that next time. So just to summarize what I've shared today, we've seen that exogenously applied double-stranded RNA can be used to inhibit the infection physiology of rust fungi. It can prevent and cure myrtle rust, and it can be used to uncover myrtle rust effector biology, amongst potentially other aspects of the fundamental biology as well. So that is everything that I have to share today. So yeah, thanks so much to all my collaborators, and huge thank you to my supervisors. I have the most awesome supervisors, so I'm very grateful to them. And yeah, thanks for having me, and happy to take any questions, and I'll hand back over to Anne.
Thanks, Beck. That was a really nice talk. We have a few questions, and we still have a little bit of time, so I might just read out a couple of them for you to answer. So the first question is, hi, Beck. Thanks for your presentation. Really exciting that infections can be cured post-infection by up to 14 days. Do you have any thoughts about how this might translate to be applied in industry? For example, spraying of rust-susceptible species in native nurseries. Have any industry partners demonstrated interest?
Yeah, so that's a good question, and you pretty much answered it. So yeah, basically we kind of foresee at least the first stage of applications to be things like nurseries and plantations, and also ex situ conservation as well. So a big part of myrtle rust management is trying to kind of grow up slightly more resistant genotypes and then distribute them, but often they still need to use fungicides to actually get these plants to a decent age. So potentially it could replace uses like that. And yeah, absolutely in native nurseries, and also like hopefully in lemon myrtle plantations or eucalyptus plantations, maybe things like that. And yeah, we do regularly get quite a lot of interest. We've had a lot of just kind of growers and also hobbyists or people who have kind of their personally owned nurseries and things like that contact us, and often they're really keen to kind of set up field trials and things like that. So that's definitely a next step. Yeah.
Yeah, and I also have some industry partners who are supporting my fellowship. So Australian Native Products, which are the largest lemon myrtle growers in Australia, are supporting our upcoming field trial. So hopefully that will go well. Okay, so another question we have is there a risk of selecting for myrtle rust uridinospores that do not take up the double-stranded RNA, thus creating a rust line that is resistant to this method of disease control?
Oh, I haven't heard this question before, but from what we've seen, all Uridinia spores seem to pretty consistently take up the double-stranded RNA. It's really, we only see that the Telia spores don't take up the double-stranded RNA, but they're not actually the spore stage that is going out and infecting. And in terms of developing resistance, I hope this answers your question, but the targets that we've chosen are actually essential barcoding genes. So it would be very, very difficult for the pathogen to kind of mutate those genes, and the pathogen would not be able to survive. So yeah, resistance arising is pretty unlikely, but if it did, the cool thing about double-stranded RNA is that it's actually pretty easy to kind of modify a construct and actually redesign it to overcome that resistance. It's not like a 15-year process like breeding a perennial tree.
Yeah, so I guess, yeah, there is the possibility that somehow there would be some mutant that wouldn't be able to take up the double-stranded RNA, but I'm not sure we'd be applying a high enough selection pressure for that. But yeah, you never know. Okay, so another question is, great talk. Do you have any idea on how the processing of the long double-stranded RNA occurs after delivery onto the plant? Is it by the plant endogenous machinery or by the pathogen?
So in this particular system, we think that the fungus is actually taking up the double-stranded RNA directly from the leaf surface. So there's no pre-processing happening within the plant. That seems to be the case in other systems, but yeah, double-stranded RNA is not actually taken, or is not easy to actually get into Myrtaceae plants because they have this very thick leaf cuticle. So in the case of this system, the spores are actually just taking up the double-stranded RNA and processing it directly. Yeah.
Okay, and we have one final question. Do you have an approximation of the costs of using double-stranded RNA for disease treatment relative to other control measures?
Yeah, so this is something that I don't know a great deal about because I'm not working heaps on the super applied end. But from what I've read about similar kind of control measures in double-stranded RNA is that it becomes much more cost effective once it's scaled up. So there are some companies that synthesize double-stranded RNA really large scale, and at that larger scale, it becomes cheaper than traditional fungicides and things like that. But at the scale that we're working at now, obviously in the lab, synthesizing all the double-stranded RNA ourselves, it is pretty costly, but not extremely so, but it definitely gets a lot cheaper as it's scaled, which is the important part, I guess.
Yeah, and there are actually large field trials going at the moment with double-stranded RNA, so it is feasible. And there is a product already registered in the US against Colorado potato beetle. So obviously it's feasible for that to go ahead. So hopefully it'll be the case for myrtle rust when we get to that point. Okay, great. So that's all the questions. So thanks again, Beck. If you just want to go to the next slide, this is an ongoing seminar series. So if you would like to sign up to hear more coffee seminars, you can scan that QR code and then you'll receive all of the information about the upcoming coffee seminars. So thank you again for attending our seminar today, and hopefully we'll see you next time.
Thanks, everyone. Bye.